Emanuela Bonanno from Columbia University. I'm here today with Christin Glowa. Please, Christin, introduce yourself. Where do you work?
I'm Christin Glowa. I'm from Heidelberg in Germany, and I'm working in the German Cancer Research Center in the Department of Medical Physics and Radiation Oncology. And my working group is named Applied Physics in Radiation Oncology. And we are a very diverse group with a lot of physicists and 2 biologists. And I'm one of these biologists.
I see. Yeah. And what's your line of work, or what are you working on these days?
So I'm focusing on carbon-ion radiotherapy because we have this unique facility in Heidelberg with the one gantry which is clinically used in the world, and we have 3 patient treatment rooms and another experimental room where we can irradiate animals or cells at night. Yeah.
So it's totally dedicated to research.
Yeah, it's for clinical research and also for laboratory research or animal research.
I see.
Yeah.
So you said it's carbon ions, so must be related to radiotherapy treatments, right? What's your model and what you are looking at?
So at the moment we are really using carbon ions, but we are also able to use protons and heavier ions like oxygen ions.
That's interesting.
And at the moment I'm working with a prostate cancer model, which is a very unique system because it's established on rats, syngeneic, so this means that we really can work with a complete immune system, which is not that normal in the radiation therapy treatment society, because normally they are using human tumors which are transplanted on nude mice or rats, and then you don't have the complete immune system. So that's a big advantage of our studies. And the second big thing about these tumor models is that they consist of different sublines. So they are originated from one tumor subline. This is the R3327 Dunning prostate tumor. And then they were transplanted sequentially of the animals. And so we get different sublines with different characteristics, like different growth rates, different differentiations, so how the vessels look like and how they benign or malignant then they are, and how radioresistant they are, because they are also different in the hypoxic status. And so we have a whole variety which is also seen in the clinic, and we can irradiate them and then see which tumors are most beneficial for carbon ion.
Well, you were mentioning that you have access to carbon beam, proton beams, and oxygen. So what's the advantage of using these particles compared to like traditional or conventional radiotherapy treatments with X-rays or?
So there is known since many years that there are 2 main advantages. So on the one hand, there is the physical advantage of the unique dose profile of these particles, because normal X-rays, they deposit their energy over a wide range, and the most energy near the entrance region, so below the skin. And the particles are deposit nearly all of their energies in a deep manner, dependent on their energy, so you can reach deep-seated tumors. And you are also able to spare the normal tissue, which is proximal to this energy deposition Bragg peak. And this is very unique to get more dose in the tumor and to spare the normal tissue around the tumor.
Yeah. And so avoid any severe late effect, right? So why are you, well, we know that there are some results from Japan about carbon ion and Germany as well. Why are you guys trying to look at the advantages that other particles, such as oxygen, for example?
So for carbon ions, it is at least assumed in cell culture that they are especially good for hypoxic tumors. And this is because of the high LET, and because you have these densely ionizing track structures where you can induce very complex DNA double-strand breaks, and they are very difficult to repair and lead to more cell kill. And this is at least very good to kill hypoxic cells, because they are not that easy to kill if because they do not fix the damage in the DNA when oxygen is not present. And if you go to higher LETs like oxygen ions, it would be assumed to be even more effective, but it's not shown at the moment in vivo. It's just like cell culture experiments, and so we are now establishing data sets for carbon ions and oxygen ions with different in vivo models, which will be then a unique data set which you really can translate into the clinical trials.
Well, I know that you can work a little bit on the energy of these ions and then try to get a Bragg peak, which is a little bit like a little bit larger than the what we think is the Bragg peak. And in that way, you can really try to target a tumor, which is not like like a pencil. Yeah. So do you have these capabilities there?
Yeah. So we superimpose Bragg peaks with different energies to get a so-called spread out Bragg peak. And in our case, we are treating tumors which are 10 millimeters in diameter, and our Bragg peak is then 20 millimeters in diameter, so that we have enough margin to really cover the complete tumor volume. Yeah, and don't have these fall-off effects at the end of at the distal edge of the Bragg peak.
An exit dose.
So that we really have a complete homogeneous dose distribution through the tumor volume.
So tell me more in practical terms, what's your experiment is about? What do you look at? What are your biological endpoints?
So I am transplanting these tumor fragments, which are originated from these typical or this special rat, which is the Copenhagen rat. And this is a very originated normal rat, so it's no typical laboratory rat where you have like many gene deletions or something. So it's a very it's a very immune
it's not immune compromised.
Right? It's not immune compromised. So it's and we transplant then different kinds of tumors. And then they grow, dependent on their volume doubling time. And when they are 10 millimeter in diameter, we irradiate them with either photons or carbon ions, because photons is always
your reference radiation.
Yeah. And our biological endpoint is local tumor control, so that we are really near at the clinics after 300 days. So it's about 1 year observation time after irradiation.
Those are long studies.
Yeah. Right. And then we do dose response studies, so irradiating different dose levels with normally about 6 animals per dose level, so that we really can have the percentage of local controlled tumors per dose level, and then calculate the TCT50 value, so the dose which is applied to control 50% of the tumors.
And do you have already, because those are very long studies, do you have already some results that you can share?
Yeah, so we have completed the results with 1 fraction irradiation. This is already
with carbon which
ions.
Energy and LET?
The average LET is 75 keV per micron. Yeah. And with 2 fractions, it is also finished, but it's not published yet. And at the moment, we are in the observation time of 6 fraction irradiation. Yeah, with these 3 different sublines, which are varying in their differentiation and hypoxic status. Yeah.
I see. So I guess your next step in your research is really trying to characterize many different ions or different LETs for your animal studies. Yeah. I was curious because you started saying that where you work, there are physicists, there are biologists. How is it for you being able to work with physicists and biologists in the same lab. I mean, as a radiobiologist, I always worked with physicists and biologists at the same time. Is it easy for you to, talk to physicists and let them know what you're looking for and the rationale of your work? And at the same time, are they open to, try different things for biological research?
Yeah, so our physicists are very open, but yeah, you really have to talk a lot with them, because sometimes they are not that deep in biology, so you really have to think about how to explain.
Or at the same time, biologists want something that physics is not supporting.
And they always complain that biology is so complex.
Or things don't work all the time the same. Yes.
Yeah, but it's in this field, I think it's very necessary to have both directions, and you need physicists, because you're not able to do everything on your own, and you need physicists to adapt the plan and also to do the dosimetry,
which is so critical. Yeah, absolutely. Christine, do you want to cover something else that we didn't talk about? Anything about your results or your future plans?
Yes, I can give you some results. So what we at the moment see for one fraction irradiation, so for single dose irradiation, that it is really beneficial in very effective in fast-growing undifferentiated tumors, where the hypoxia is big, because there we get the largest RBE, and this is about 2.3 at the moment for carbon ions.
Wow. And again, at 75 kV per micron, 2.5 RBE, 2.3 RBE.
And there is a slow increase if you go to 2 or 6 fractions, which is about 2.4 or at least 2.5 for 6 fractions. Yeah, and if you go to more differentiated tumors, then the RBE decreases. For the HI tumor, it's about 2.1, and then for the H tumor, which is very benign, it decreases to 1.6. But this is not due to the carbon ions. This is due to the photon dose response curve, which is our reference. Because for the benign tumors, you can really cure them good with photons. And so you only have like a shift of the photon dose response curve, and the carbon dose response curve nearly stayed at the same dose, and therefore you get these lower RBE values. But the very interesting thing about this is that it's really true. Like you have some threshold where you irradiate with carbon ions, and then it's not that dependent on tumor intrinsic factors like hypoxia or the intratumoral heterogeneity. You are able to cure tumors. And the second thing, which was very interesting for us, is that when you irradiate very heterogeneous tumors with photons, you get a very shallow dose response curve. And if we do the same with carbon ions, we get a steep dose response curve. So that there is really, that there is really the conclusion that you are less dependent on these tumor resistance factors and are also on tumor heterogeneity, which would be very beneficial for clinical treatment.
For clinical treatment, particularly of tumors that are very difficult to treat, and don't respond very well to conventional radiotherapy. Just another question. You say that you work with the prostate cancer model, but I know that you are also involved or collaborate for spinal cord irradiation. Can you just tell us a little bit about that model too?
Yeah, so in our group there are 2 biologists. One of them is, I'm the biologist, I'm working with the tumor model, and there's another postdoc, which is Maria Sager. She's working with the normal tissue, and she is mainly interested in spinal cord and different fractionation schemes and how the what's the word? So the injury of the spinal cord is dependent on the different LETs, so between the plateau and the different energies at the spread out brag peak and before the spread out brag peak, to really see the LET dependence of the injury.
I see.
In normal tissue.
And she uses the same model. It's a rat model.
It's a rat model, but it's a Spagdoli rat.
Okay.
Yeah, which was
more specific for.
Which is it's used for spinal cord irradiation since I think about 30 years.
I see.
Yeah,
so
which was established from Albert van der Kogel.
Mm-hmm. I see.
Yeah, decades before, and so you have like literature values for everything. You can compare it with different X-ray schedules and so on. Yeah.
Okay, so anything else that you want to add? No? Then I thank you very much for taking the time to talk to us for the Vodcast. And I'm looking forward to read more about your studies, and good luck.
Thank you very much.
With your work.
Yeah.
Thank you for your time.